thank you so much for being here. I am hoping that I will inspire you about plankton. You may or may not know what plankton is. Oh, there we go. Just a short bit about me, not that you're going to read this, but I started in high school a long, long time ago. I read, Mind in the Waters about dolphins and realized how amazing the cetaceans were, and that they were, at that time, in the 70s, being killed on a large basis, and so saved the whales, did dance-a-thons, and had a chance first year in college to go to Hawaii and study humpbacks, and transferred to UCSC to work with Ken Norris on humpbacks, or on whales. But I found that at that time, the folks that were doing that research didn't play well together, and I just got really down, really like my dream, my passion for the whales was kind of torn apart, and I thought, I need to do something else. And John Pierce at UCSC, if you know the Pierces, totally inspired me about invertebrates and the critters, in the mud. And every summer growing up, I would go to Maine. We lived in Davis, and my dad's family was in Maine, and I realized that it was that got me totally inspired and hooked on plankton. Because how many of you have ever seen bioluminescence in the water? Oh, wow, that's fantastic. It's magic, right? I mean, my cousin and I would sneak out and swim out to our friend's boat, and, each finger would be lit up with the bioluminescence. And so, that inspired me to study the critters in the mud, and so the rest is kind of history. I met my husband in the mud, Mark, and we had the twins, and those twins that Christine helped raise are now don't look anything alike, even though they are identical twins. I know, but I am incredibly, incredibly lucky to teach a class at Cabrillo that is no credit, no fee. It was created by Nicole Crane and Lisa Utall. That basically we go out, we collect at 4 different places, we bring those samples back, we identify them, and then we enter them into a national database, NCOS, that is critical to, a lot of professionals are doing this as well, but we are the citizen science group that is doing it. And just the T-shirts are all their design. Yeah, and so that's been great. But truly, to me, the reason that class exists and all the fabulous research around the world that is being done on plankton centers mainly on, what are called HABs, harmful algal blooms. And it is true that it is absolutely critical to monitor these, to let people know when a bloom is happening. And, the reason we don't eat clams, mussels, any kind of filter feeding marine organism during the summer months is because that's when the dinoflagellates and diatoms, tend to bloom, and that can, kill definitely animals, marine animals, and people. And so, but I tend to like to focus on the beauty and diversity. I have focused most of my time at the Monterey Bay Aquarium on 3 different exhibits of plankton showing the animal plankton. And so that, was my strong suit, are more the animal plankton. But truly what I've come to realize is that it is, when you look out at the ocean, you look out at the water and you don't see anything, right? I mean, this is Whale Fest, and so I'm sure all of you probably have gone out on the boats or even from shore and seen dolphins, seen the blow of the whale, and that's fabulous. But what we're talking about are the microscopic, tiny organisms that are within a drop of water. And so, looking at a single drop of water, no exaggeration, can be filled with, hundreds of animals and literally thousands of phytoplankton, the plant plankton, and we're now realizing that the bacteria and the viruses and these weird things called archaea are even more abundant and important. So we keep learning more and more. But it's like, the night sky. If you don't have a telescope to look up and see the details, you know very little. Same thing with the plankton. And so, even Cahill Gibran that gave us, at least I see many people my age, so if you took part in any weddings, you probably read from Cahill Gibran if you're like me. And I love this, that in one drop of water are found all the secrets of the endless oceans. And it's true that almost all of the organisms that live in the ocean start out as plankton. And so, actually, I'm going to skip to this one. So what is plankton? Plankton comes from the word planktos, so the analogy or metaphor of, the planets rotating around the universe or moving around the universe is pretty apt because it's any organism that is at the mercy of the major ocean currents, right? How many of you have ever been plankton? Yep. So I was like 16 and I was surfing I wasn't really surfing. I was trying to learn to surf out of Bolinas, and I was in my friend who was 6'2"'s wetsuit, so I was not very warm. And I'm like, I'm going in, and I started crossing over from Stinson Beach to Bolinas, and there was a rip current that just took me out. And so we too can be plankton. It just means that we're at the mercy of the currents. All of these organisms do move. It's just that they can't move, against the major ocean currents. And the amazing thing to me is that now with all the satellites, technology, we can literally see these plankton blooms from space. So all of the green that you see in the little whorls are plant plankton, phytoplankton, probably from the color, diatoms primarily, but hard to say. But they are so large and so vast. And so I made the reasons why we should appreciate, the plankton. One of them is that at least 30 to 50% of the oxygen we breathe is from these phytoplankton. In schooling, how many of you ever heard that? That, half the oxygen we breathe. Oh, good. Some younger folks, definitely got that. That's great. Because I know I didn't. And, the number varies so widely because it depends on what your focus is. Some scientists are like, well, anyway, it's a complex thing. But we know that much of the oxygen we breathe is from plankton. And of course, the number 2 reason why plankton should be revered is that it is the very base of the food web in the oceans. And so the phytoplankton feed the animal plankton, the zooplankton feed the foraging fish. I know on the Santa Cruz side, I do live in Santa Cruz area, Aptos, we've been seeing, probably not for the last month, but before that, for at least 4 months, on and off, the anchovies in large numbers, the sardines coming in. what we're finding is that, and I'm sure maybe some of the other speakers talked about this, that the humpback whales that feed on both krill and the foraging fish, will follow these foraging fish inshore. And that's why, there's been this problem with them getting caught up in the crab traps, and why the crab fishing, has been stopped at certain times. With climate change and things moving closer to shore, things are definitely changing. Basically the same thing, just showing, the importance. And I'm sure you probably, I can see that this is an educated group, that you probably know this very well, but for those that don't, we are one of the few hotspots around the world, and the reason for that is that we are an upwelling center. And so the green is showing, types of upwelling, the seasonal upwelling, which is what's happening right now. The winds that we had yesterday, and, as long as we have northwesterly winds that are continuous enough, to really drive the upwelling, we will get these bursts of deep nutrient-rich water that are, creating a, nutrient-rich area. And so when you look at this map, you can definitely see that it's areas on the eastern sides, usually, of the continents, but not always. There are some exceptions. It's hard to really show upwelling, but in our area, it does happen to the north of Monterey Bay, in Año Nuevo and north of there, and to the south in the Point Sur area. And it's all about the geography and how the points are kind of coming out. And what happens is the wind blows and blows, and because we are a rotating Earth, the Coriolis force pushes whatever, whatever, is flowing this way, which is pushing the water, and then we are rotating, so 90-degree angle from that goes this way, so the surface water gets moved offshore, which causes that cool, nutrient-rich water from the continental shelf and below to come up. And all year long, all that life that's been created on the surface has come down and settled on that floor. So it's literally like adding Miracle-Gro to the ocean. Early on at the aquarium, when we had a El Nino, a really warm season, and the kelp was really sad in the kelp forest exhibit, we actually did an experiment of adding Miracle-Gro to the kelp forest exhibit, and lo and behold, the kelp came back. That was husband number one that did that study. We won't talk about that. But yeah, so you can see the just wind, upwelling. So very important part of our area. And I'm going to hope that this works. This shows my my fabulous students at work, and just, how do we collect plankton? Plankton is microscopic. It is very spread out, and so we use plankton nets. And some of my students are here. Yay, great to see you all. And so let's see, this is a movie that Tanya McLean did. Let's see. Yeah, I know. Let's see, maybe if I do. Dan, any thoughts?
Well, there's a sign at the bottom that says cannot play media.
Oh, is there?
Yeah.
Sorry, I was outside for a second.
That's okay. What do you think? Is there?
Oh dear.
Yeah. If it doesn't.
There's nothing on the bottom. Here's the.
Yeah, there's that. That's okay. That's all right. I don't want to.
Darn,
Yes,
sorry.
that's all right. Thank you. But it shows us basically, anyone that collects plankton, actually, I shouldn't say that, researchers go out on ships or small boats and have large nets that they tow behind the boat, and that really concentrates it. But what we do is every Friday we go out to, Santa Cruz Wharf, Capitola, Seacliff when it was there, the harbor, and we have a zooplankton net that has a wide opening and is about 10 feet long, and it's fine silk, and then a phytoplankton net that's smaller and has 20 micron, opening, so very small. Let's see, there we go. And so it just, life happens. Yesterday, we were here all day with the Pacific Plankton Program, and we had samples from Monterey Wharf. And over the course of 30 years of sampling plankton, whenever I come to Monterey side and I do a plankton tow, it is so rich and so diverse. And so I was so excited about, we had many microscopes set up, and the day before in Santa Cruz, we did the tows, and it was upwelling. It was the incredibly diverse, diatoms and probably 30 different types of animal plankton. Anyway, yesterday, this is what we had. The entire sample was chocked full of akashiwo. We have a bloom going on right now that is very concentrated. It seems limited to, Monterey area. I looked for a photo of the kelp forest exhibit right now, but I think if you go there, you'll see that it's like split pea soup. And that's because the akashiwo is very small, and it is it's just coming right through the filters at the aquarium. And George Matsumoto, Dr. Matsumoto, dove in the exhibit on Friday. He said he could only see like a foot. And so this is what happens, but it's unusual in that this dinoflagellate, we have diatoms and dinoflagellates in terms of the phytoplankton, the plant plankton. And this particular one usually is very abundant in later summer and fall when the water is really warm. That is the archetypical, seasonality of it. But birds have been, washing up. Akashiwo is not a toxic dinoflagellate in the sense of, like, the pseudonytzia is the one that's been killing sea lions down south and in various areas. And we had a lot of pseudonytzia, which is a diatom, in Santa Cruz on Friday. But anyway, it is the reason that it affects the birds. It produces a surfactant that is like it strips the oils from the feathers of the birds so that they can't, their water protection goes away. And so, that's its magic power. And so, sorry. I think the thing that I just want to get across is, just like on land, we have the winter, spring, summer, and fall. Around here, we don't have as much of that diversity. Is anyone from the East Coast, where you have a real fall, and winter? Here in Monterey Bay, there's this fabulous website called Seasons in the Sea. And you can go to any one of the months and you can see what's happening in the bay, what whales tend to be here, what the oceanic conditions are. And so right now is upwelling season. It's the time when we usually have the northwesterly winds causing the upwelling, and we usually have a huge number of diatoms, which causes the zooplankton to bloom. You can go to Embari's website, and you can go to the, either the CENCOS, it's the Central Northern California Sensing Center, and it shows the surface temperature mapping, where you can see the different years, and this isn't the latest one, but I just wanted to show, where it's, January, February, March, April, May, June, all of these months are very cold water, right, on the surface because of the upwelling. And that's all I'll say about that. Any questions about that? Yes?
How do you spell pseudo?
Oh, thank you. Yes. So it's P-S-E-U, let's see, U-D-O, pseudo, and then Nitsia, N-I-T-S-C-H-I-A. It's, yeah. Yeah. I might have said that wrong. I'm terrible at spelling out loud, but I can write it down. Yes. And I'm terrible at timing too. What's my timing like? Okay. Okay, good. Good, good, good. Okay, good. Awesome. And so, obviously, all plankton is not the same. Plankton just means that you're a drifter, right? So we could be plankton, like I talked about. Jellies, those beautiful animals at the Monterey Bay Aquarium and other aquariums, are drifters. They are, very large. In north, up in Alaska, they can be 50 feet long. The plankton that I'm talking about is microscopic. And so we have the dinoflagellates and we have the diatoms that are the phytoplankton, those that are using the sun's energy to produce, carbon and are food for all the other animals, or zooplankton. And just this is a scanning electron micrograph picture of a dinoflagellate, and you can see that the girdle here and there, and they move very quickly because they have a little flagellum, and that makes them different from diatoms. And so, like I said, the animals are the zooplankton, and the phytoplankton are the plants. Many of these photos are Christian Sardet. If you ever have a chance to either, get his book or, just go online and look at his work, it is absolutely spectacular. He and his son, Christian and Noe, came and helped us put together the exhibit on plankton at the Monterey Bay Aquarium. So those beautiful images are Christian Sardet's, S-A-R-D-E-T. And so, believe it or not, these organisms, of course, the copepod known from SpongeBob, all the kids yesterday, I was so excited, new SpongeBob, which I wasn't sure about anymore. So, the copepod, but, organisms like this that I think are just incredibly gorgeous become a tiny little worm-like animal that is not very attractive, but as a plankton is amazing. And this amazing being is actually a relative of a shrimp that is a amphipod, and it was what they used as the model for what's it called, Christine? Alien. Thank you. Yes, that was Alien. The guy who made that movie was a marine biologist. One of them.
You can't tell me that George Lucas gets to.
What's that? Yeah, right? Yeah, it's true. What's that? That's right. Thank you. The phytoplankton, comes in so many different colors. Right now, they're all golden, but it's really amazing that we use a chart each Friday when we go out, and we say what number on the chart is the water today. And each of these organisms have different chloroplasts, just like plants have the different chloroplasts, and it makes the water different colors. But truly, to me, it's just amazing that, like on the far right over there, a chain diatom that is the shape of a Japanese fan. These things, you just can't make it up. And so plankton is freshwater and saltwater. Plankton in the freshwater community is the base of the, freshwater community. And it can be a 100-foot-long siphonophore in the deep sea, or it can be, the tiniest little bacteria. And the other way to classify them is those that spend their whole life in the plankton, holoplankton, like our friends the copepods, the krill, the jellies, the medusa stage of the jelly, and then the meroplankton. Every single animal you've ever seen in a tide pool, almost every animal, there's a few exceptions, have their larvae as plankton, called meroplankton. And the other reason to appreciate plankton is the fact that, the diatoms have their houses or their bodies are made of silica or glass. And so when you look at, just on Highway 101 over in Salinas area, the Dolomite Mountain there is the ancient remains of the diatoms from millions of years ago that have compressed over time, and then those uplifted, because we're in an uplifting zone here, and then it is harvested and used, for pool filters, for I just learned the head on a beer, and for a lot of different industries that need that fine silicate. Also, the paint on our highways, how at night sometimes it glistens, because imagine if it's crushed glass, when lights hit it, it diffracts those different colors. So I'm not going to play this video because I know it won't work, but if you're interested, Journey to the Microcosmos is the most fabulous website, and I highly recommend it. There we go. And our little friends, the coccolithophores, which in my bag I have a model of one, and that afterwards, if anyone wants to look at it, I'm happy to show you. They are incredible, and it is a type of, phytoplankton, but they're actually like an amoeba, that they're protists, and they have calcium carbonate, quote unquote, shells or outer, structure. And the White Cliffs of Dover are coccolithophores. Our friend that helps with the class calculated how many it took, but I don't know. Any of you, do you remember what Clive estimated it to be? 100 million billion. And even here in Monterey Bay, we occasionally get coccolithophores. Coccolithophores tend to be more warm water, and but in 2015, does anyone remember when the water just on the Monterey side looked like this? It was amazing, and it was a bloom of these little beings. And like I said, you can see it from space when they are really abundant. So, looking more closely at some of these, like dinoflagellates. So, like there's the diatoms and the dinoflagellates. So, kind of easy to remember. Dino to flagellates. So, they all have one, kind of like our belts. They have one flagellum there, and then another one along the side. They're amazing in their adaptations for life in the ocean. This is a Christian Sardet image where, we've seen these over the course of umpteen number of years. And there's different species sorry. What he found is if the light, let's say the sun, clouds come over and the sun is not strong, instead of, suffering, they literally create fingers and increase the surface area for the absorption of sun for photosynthesis. And then when the sun comes out again, in they go. So just amazing with their adaptations. The jellies that we know and love, definitely it's the Medusa phase that we see as plankton. They do have an attached stage that is not plankton. Like I said, plankton just means you're a drifter. It's really a catch-all term. But I wanted to just, showcase that. And right now, how many of you have seen this? All right, yay. You probably, many of you know what it is, but I love By the Wind Sailors, Villela Villela. And, last year they came in humongous numbers. I heard that it's at Asilomar, Moss Landing. I saw them in huge numbers for this picture. At the end of 41st Avenue in Santa Cruz. And you can see the size by my tennis shoe. The cool thing about them is that they are made to be out in the open ocean. They have a sail. They're colonial animals. So and each individual is made up of thousands of individuals. One individual is the sail. One individual is the base, and then I'll show you underneath, all the feeding individuals. And they have their sail set at 45 degrees so that in the northern hemisphere, we have the gyre going this way so that it keeps them in the open ocean most of the time. But when the wind gets wrong, they come ashore. And, it's just part of their life cycle. So this is what they actually are. There's thousands of individuals with different mouths and thousands of individuals with stinging cells. But if you pick one up, you don't have to worry about it. Don't put it on your tongue or your dog's tongue because it does have stinging cells. Even like if it's dried up, as long as the blue part is still there, if you add water, they can still, sting, tiny things and sensitive things like your tongue, which I know that sounds weird, but people have done it. And then this is just a better picture, that you can see the golden color on the top. Imagine these by the millions on the surface of the ocean. And these are the symbiotic zooxanthellae, or algae, that are living in their tissues. And then all those individuals underneath are the feeding and reproductive individuals. So just amazing to have a chance to see them. And, they'll go away soon. But I wanted to just talk a little bit about the life cycles of, the things that we see a lot of in our samples, and that there's classes at Hopkins looking at the life cycles of these various organisms. These are the tiny snails that you see right at the edge of the rocky shore. They are unusual in that they'll come inshore, they'll mate, and then the female will lay this beautiful egg case. And within the egg case, there's all these pink spots. And those each become a free-swimming little, what's called a veliger larva. And then it'll be in the water, swimming, eating for several weeks, and then it'll settle down and become a good little snail. Many of you have probably seen kelp or other seaweeds that look like this. Anyone? Yeah. And so, I've always been curious about those. Kind of looks like dried salt, right? But it's actually one of the most beautiful, I think, organisms called a bryozoan. and it's in a whole phylum all to itself. And they have a larvae that looks like a little party cap. And it's, 2 weeks ago we had a lot of these. And so they're swimming around, and I happened to catch this sequence several years ago under the microscope, so I was really excited. So that you see, this is the larvae that the organism actually, well, I'll tell this first. So it swims around, and then it changes shape from, the flat to round, and then when it settles, it immediately becomes two individuals, or twins. So I have this strong connection with the bryozoan because it too has twins. And then it divides and divides into, many individuals, and they, the kelp blade doesn't live very long, right? And so they have to live their life very fast. And then the kelp blade dissolves, and that's the end of their life. So very, very short-lived. But barnacles, if anyone, does anyone have a boat in here? No? Yeah, kind of. Yeah, people that have boats do not like barnacles, I have to say. They do attach to anything and everything in the ocean, including our friends, the gray whales. Interesting that the nauplius, the little first larval stage of the barnacle, it will only settle on whatever species it is meant to settle on. So in the Baja lagoons, the gray whale, this is the time when the gray whales are getting their barnacles, because it goes from this stage to this stage to this stage, and when it looks like this, it is not feeding any longer. It has these appendages and an eye spot and legs for moving around, and it has these fine little hairs, and it literally tastes the substrate, and it has to have eau de barnacle in order to settle, because I don't have a picture of it because we could have kids in the audience. The barnacles are known for, they actually need to settle next to each other because they're one of the few invertebrates, marine invertebrates, that mate rather than sending their gametes into the water. And so barnacles are known for having the longest male organ because it has to reach to the other barnacles in the area. I saw it once under a microscope when I was setting up the kelp lab at the aquarium. Oh my gosh, it was amazing. Yeah, okay, thank you. Julie Packard was with her young twins, and I don't think that she appreciated me calling them over to look. Okay, so, just sharing the fact that, to be a drifter, you need to be transparent so that you hide, so that your predators, which are everyone, don't see you as easily. We see a lot of organisms with long, thin spines, so that it's, kind of like not great to take a bite into. And they have amazing different ways of keeping buoyant to keep up towards the surface of the water where all the phytoplankton are. And so you'll see oil droplets and even specialized metabolic system just to stay buoyant. But as I mentioned at the beginning, we do have those problem critters that are, the ones that have the red tide. And not all red tides are toxic, but many of them are. And so the red tide, is what I was talking about earlier about the HABs, or harmful algal blooms. And the group at UC Santa Cruz, Rafael Kudella's lab, studies those, and they have amazing ways of sampling all throughout the ocean. That said, and I think you all probably know this, but one of the things that I'm most worried about right now is the fact that NOAA are the people that are out there studying all of these organisms, which means when there's a toxic bloom, they're going to notify us. They're going to notify everyone that needs to know. And what, 60% have just been laid off? And so, we really need to, I don't know what, protest and, but yes, talk to your representatives, absolutely, because it's happening right now, and we need to definitely, yeah. So one of the last things is that while we are asleep at night, these amazing organisms are moving from the depths several hundred feet down, and they migrate up towards the surface of the water. It is the largest migration on planet Earth, and almost no one knows about it. It is the diel vertical migration. Not all the organisms take place in it, this happens with, but a lot of them do. It was first discovered with World War II in the submarines and the advent of the echo sounders. And so just incredible. One of the big ones that does this are the copepods, and that, at night they're down below. Daytime they're down below, hiding from predators. During the nighttime, they come up to the surface to feed. And they are some of the most amazing grazers. There's a gal that years ago at NPS did a study where she glued a live copepod, calanoid copepod like this, just the carapace, super glued it to a dog hair, and then had a stem into a aquarium, and then had a high definition camera, and she was able to record the feeding of the copepods. And it was just incredible because a single species of copepod can graze out the phytoplankton in an area in just a night, in a limited area. So they are truly remarkable. If I ever get a tattoo, which I probably won't, this is what I would get. It is the most amazing zooplankton larvae I've ever seen, and it's a weird little animal that, almost no one knows about. And it looks kind of like an octopus squished between two shells, and it's an ostracod. I'm sorry, not an ostracod. It is a lingula larvae. So the lingula is just this weird animal. So I guess the bottom line is keep your eyes out for anything, unique happening. These organisms, like the coccolithophores, are definitely at risk of ocean acidification that is happening right now due to the, rampant carbon dioxide due to our, a modern way of life that you all know about, I'm sure. But numerous studies are being done all throughout the different universities documenting, these changes that are happening. One good thing is that Monterey Bay is, an upwelling center. And so what we've found is that the water from down below is naturally more acidic, and so the organisms here are a little bit more adapted to those changes. But, and then also just to know, the microplastics that we are all trying to keep out of our lives and keeping out of the ocean, not only is it, in the upper region of the water. They did a study at Mbari and the aquarium with these squat lobsters that came in during an El Nino. There were millions of them. And so it was a chance to look at the stomachs and to see, what are they eating? And is there any microplastics within them? They found a huge amount of microplastics, and that's what led Mbari to do the study in the deep waters, and they found the same thing. So, the things that we need to really work on, I think hopefully sharing the beauty, diversity, and if you have a chance to look at the microscopic world, no matter where you are, like in your backyard. There's at Stanford they have the foldoscopes for like a couple of dollars. You can make your own little microscope, and you can attach it to your phone, and you can take a little sample, put it on there, and look. So you don't necessarily need the expensive, equipment, and you can take a look at what's going on. So thank you so much for coming to my talk.